is produced, and the protein difference can change the way it binds to the drug. So that's how a SNP gets converted to a mutation that has a biologic change. So this gene has been studied over and over and over and over again in Caucasians. And we're one of the first to look at it in Latino populations. There have been several SNPs. There's about 13 SNPs, and we looked at all 13 of them. One of the SNPs results in an amino acid change, and I've showed that here with R's going to glycine. And what we did is we compared, at least in the Puerto Rican population, we compared genotype status, whether you had two arginines, an argine and a gly, or a gly and a gly, to drug response. Remember, the higher the better on the y-axis. And what you see is, at least in the Puerto Rican population, a very nice dose-response curve. The more arginines you have, the better your drug response. The more glycines you have, the worse your drug response. But what we also found was that there was a strong interaction between that genotype and asthma severity. So amongst kids that had the most severe asthma, the effects are much, much more pronounced. Huge effect if you had arginine, negligible response rate if you had glycine. So the important point, though, that you don't get from this slide is that these two bars make up 80% of the Puerto Rican children that we tested. Out of 386, 80%. So that's a substantial proportion of the Puerto Rican population that is walking around carrying a gene that has confers low drug responsiveness. So again, tremendous public health, tremendous potential for political implications, but they're the wrong population, so most people don't care. But we didn't see that same effect at all in Mexicans. Nothing. So this is a challenge. This is a challenge because we ask ourselves what's going on. And this underscores, whenever there's a challenge, it's a challenge to win. It's a challenge to try to better understand what are some of the environmental factors, what are some of the dietary factors, what are some of the gene-gene interactions, what are some of the genetic, what we call epistatic modifiers that are somehow modifying this effect in the Mexican population that we're not seeing in the Puerto Rican population. And just to put this in context for people that may be statistically minded, this wasn't due to small sample size. We had, remember, about 700 kids. This is huge, very big. In fact, it is the largest study, family-based study of a minority population for asthma in the world. So this is not due to small sample size. Remember that slide? This just underscores some of the complexity. So let me just tell you how we use these ancestry informative markers, or these forensic markers that I talked about. Well, we all have SNPs and genetic variations in our genome. Now, we probably all share a lot of these variants, but some variants are or occur more commonly in one population versus another population. And one way that we can measure it is by comparing the frequency in one population versus other populations. And for us to get these variants, or what we call Ames markers, we just looked at the delta difference, the difference between populations. And a poster child for this is called the Duffy locus. This is a SNP. It's not a mutation, just a genetic variant that occurs in Africans. It doesn't occur in Europeans and Native Americans. Now, you could be 100% African or look 100% African and not have the Duffy locus. I know someone like that. But the combination of several markers, not just the Duffy locus, but say 40, 50, 100 markers, can give us a pretty good estimate of that person's ancestry. And that's what we use to measure all of our ancestry. Just to show you some of the complexity of studying racially diverse populations. I mentioned the false positives. We took these ancestry, 4 of them, and just put them between cases and controls. And they have nothing to do with asthma. So what do you think the frequency difference should be between our cases and controls? There should be no difference at all. If they have nothing to do with asthma, we should see that the cases have the same frequency as the controls, if they're genetically matched. Everybody swore that they were Puerto Rican in our group, so we just did this for Puerto Ricans. And what we found was that, at least in the Puerto Ricans, 44, we got 9 that were statistically significant. So, have you guys taken statistics? Okay. If you do 20 tests, the p-value 0.05, how many are supposed to be positive? 5%. So you do 100 tests, 5 should be positive. Okay? So we did 44 tests, 5% of that, 2.2 should be positive. We got 9. This is the problem. This is why geneticists wouldn't touch racially admixed populations with a 10-foot pole. So we use these Ames markers. Huiru in my group has developed, I mean, she's a statistician. This is all she does. Sits there and thinks about mathematical models, takes these Ames markers, how to develop some of the statistics of measuring ancestry per each individual and including that as a covariate in our model, just like height, weight, and gender. So we come up with a percentage, and we could statistically correct for the racial differences between our cases and controls. And when we did that, what she was able to do, if I can get this to work, is she made almost all of them go away. So 0.007, now it's negative. And that's what you should see. So we've corrected for the racial heterogeneity. And we got 3 markers that were now associated, and that's near the 2.2 that we expected. So that's the downside. The positive side is this. Say this is the karyotype. This is called a karyotype. You guys will get this down in medical school. Say this is the karyotype of an admixed population. This is a challenge. But what if a gene, shown here by a D, originated on a yellow background? And so that everyone that has that disease, say asthma, inherited that same mutation. Well, what if you used a marker, shown there in purple, that picks up the yellow background? Now, if you had enough markers, not 40, but a couple thousand, you could be able to hone in on the signal that's emanating from this D allele, this D gene. And that's called admixture mapping. So that's one of the opportunities of studying racially admixed populations. And to explain it in terms of simple genetics or biology 101, remember when 2 populations form, blue and yellow, they meet, just say that the D originated on the yellow background, and then just say that they're random markers. The big A and big B pick up the blue background. The little a and the little b pick up the yellow background. Okay, those are called, we'll just call those the Ames markers. Through meiosis or recombination, there's crossover. And so the little b in this case is all, because it's physically close to the D gene, is always likely to ride with it. Okay? If you guys don't understand, please let me know. And then through meiosis, when you have offspring, before you have offspring, you make sperm and eggs. And just say that the D is still tracking along with the B marker. So if you wanted to find the D, if you knew a priori that the D allele was next to the B, all you have to do is find out where the B is. Okay, but just say that the B allele picks up the yellow chromosomal background. Just say in this case it's European. So you have an ancestry reporter marker that picks up European background. Okay, you have enough markers. If you could pick up all the segments of the European component, say of an African American, and hone in on a gene that may have European origin. To put this in context, to give you a good example, we know that multiple sclerosis, it's a neurologic disorder. It's a Caucasian disorder. Happens in Northern Europe. Does not happen in Africa. But we know that African Americans get multiple sclerosis. So why? The reason is that they likely inherited a Caucasian piece of the genome that is carrying the MS gene. So if we